So, hi. Today I will present a paper called Computational Analysis of the Influence of the Model Boundary Conditions on the Bacterial Cell Organization, written by me, Martinas, Mačernius, and Romas, Baronas. So let's begin by defining a few key terms, starting off with chemotaxis. So organisms are constantly affected by chemical stimuli in their environment, whether by pulling them towards them or pushing them away. Such movement is called chemotaxis. And the organism of interest for this paper is the E. coli bacteria. To be more specific, the E. coli exhibit chemotaxis to self-excreted attractant, which results in the swarming of bacteria. And what you should know for later is that cultures of E. coli are capable of self-organization, forming growth patterns of millimeter scale and exhibiting strong inhomogeneities in their density, mainly near the contact lines and surfaces. So when considering the mathematical modeling of chemotaxis, the Keller and Siegel mathematical model is the first and one of the most popular currently used models today. And according to Keller and Siegel approach, the dynamics of the bacteria and chemoattractant are mathematically modeled by a system of nonlinear equations of the reaction diffusion chemotaxis type. Now, normally, the processes in the bacterial culture are complicated and need to be modeled in a three-dimensional space. But due to the accumulation of cells near the contact line, as mentioned before, at the top surface of the fluid cultures, the essentially three-dimensional process may be approximated in one dimension. So let's take a look at the foundation of this work. A model was taken from a paper called Modeling the Bacterial Cell Organization in a Circular Container Along the Contact Line as Detected by Bioluminescence Imaging, written by Romas, Baronas, and Mačernius. And it mainly focuses on modeling the bacterial migration near a three-phase contact line with a one-dimensional in-space mathematical model, and is defined in an interval and uses a periodic boundary condition. To better illustrate that work, here is a video of an in-lab experiment using a bacterial culture. This is a view from the top of the vial that the bacteria are in. And as you can see, over time, the bacteria shift, especially along the contact lines with the surface of the vessel that they are in. And so what was done? The side view was taken into the image you see on the left, how it changes over time, and a mathematical model that attempted to replicate the pattern was made. You can see it on the right. So what was done in this study in particular? We wanted to focus on the boundary conditions. So two main boundary conditions were taken into consideration: the zero flux boundary condition, which is when the bacterial density is not passing through the boundary, and the Dirichlet boundary condition, which is when the bacterial density is held a fixed value at the boundary condition. The numerical simulation based on the governing equations of the reaction diffusion chemotaxis type was carried out using the finite difference technique. So let's take a look at the results. So starting off with the zero flux boundaries, on the left you can see the visualization of the bacterial density pattern. In the middle you see the chemoattractant concentration, and on the right is the average cell distribution. So as you can see, for the majority of the pattern, it is mostly the same, except for the boundaries, located at the x value of 0 and 1. There, the bacterial structure forms an almost straight line with few branches that are hard to see on the visualization of the structure, but on the average cell distribution graph, it can be clearly seen that the bacterial density is roughly at the same level, and then it spikes at the boundaries. Moving on to the Dirichlet boundary condition, at first a really high value was selected for the boundary condition. Here on the left is the same zero flux condition, and on the right is the high certain bacterial density Dirichlet boundary condition. And you can see that the bacteria basically avoided that part entirely, that boundary. And in the average cell distribution graph, it can be seen that there is a small point where the value is somewhat normal, that is the value of the boundary condition, and then it suddenly drops, and after some space, it returns to somewhat normal levels. So what was thought of was to use a slightly lower value for the boundary condition. So here you can see basically identical setup, except for the boundary values are closer to the background level values. And instantly you can see that the results are more similar to the zero flux boundary conditions, as once again near the boundary that is located at the x value of 1, there is a spike in average cell distribution, except that this time it's located slightly further from the boundary itself. So what kind of conclusions can we draw from all of these results? So we have shown that the numerical simulation based on the Keller-Siegel mathematical model can be used as a tool to study the formation of patterns representing the self-organization of bacteria, and application of different boundary conditions correspond to different experimental conditions. The application of zero flux boundary conditions can be used to model the vertical bacterial cell organization in the vessel, where the bacterial population tends to accumulate near the boundaries that restrict the cell movement outside the vessel. The simulated spatiotemporal patterns inside a one-dimensional thin space domain are similar to all three types of boundary conditions, only if the outer fixed bacterial density and the chemoattractant concentrations are similar to the corresponding average values. And finally, the spatiotemporal patterns change boldly when the mixed boundary conditions are notably different from the corresponding average values. Thank you for listening.